Hi, my name is Francesco Tamagnini and I work here at the University of Reading School of Pharmacy. I am a member of the Alzheimer's Research UK network for the Thames Valley. And today I will talk to you a little bit of what we do in our lab here at Reading. My group focuses on characterizing the electrical alterations and the structural alterations in the brain of mouse models which express proteins which are found in the brain of people with dementia. So we do have different approaches. We use genetically modified mice which express these proteins, or we take these proteins and we add them in the culture medium of petri dishes which contain these nerve cells, or we take sections of the brain of perfectly normal animals, keep these sections alive in a solution called artificial cerebrospinal fluid, and add these proteins to that solution so that the brain slices are exposed to it for a certain amount of time. And then what we do is to place these brain slices into a machine called electrophysiology rig, which in this case is also implemented with a specific very powerful microscope called a two-photon microscope, and that will allow us to measure the alteration in the electrical activity of single nerve cells into a complex network after they are exposed to proteins which are associated with Alzheimer's disease and dementia. Here you can see these faint, but it's actually quite crispy considering that the sample has not been stained in any way. These pear-shaped cell bodies, here you can see one a little bit better, yeah. These are neuronal bodies. If you squint, you can even see the processes going down. So all these neuronal cell bodies are in the part of the brain, the hippocampus, which is involved in the encoding of new memory. So what we do is to fill up these cell bodies with a probe, a glass pipette filled with a solution that resembles the content of the neuron. And on top of that, we have a fluorescent substance, which in a second I will show you how it will enable us to reconstruct the structure of the neuron within this complex native matrix. At the same time, this micropipette is connected to a complex electrical system which will enable us to visualize the electrical activity of this neuron live on our screen. What you see here are some pre-recorded, so to speak, wobbly lines. So what are these wobbly lines? If you get close enough to the cell, right, and then the plasma membrane of the cell gets attached to the glass pipette, and then you break the piece of plasma membrane that there is between the pipette and the cell, so the inside of the pipette is in contact with the inside of the neuron, then you will be able to measure the electrical activity of that neuron. This is a specific technique that is called voltage clamp, which allow us to measure the electrical currents that flow through the neuron. So you see here that you have these spiky-shaped electrical currents. What are they? And why do we care about that? Well, we care about this because we do know that these currents that we record from one cell are evoked by the neurotransmitter that is secreted by the presynaptic cell. So when the neurotransmitter binds the postsynaptic cell AMPA receptor, that's how it's called, sodium enters the AMPA receptor, generating an electrical current that we can measure. And what can we measure? We can measure its amplitude. We can measure the frequency of the events. As you can see, there are many events. We can measure, how fast the current is generated and also how fast the current disappears. And as a matter of fact, what we did observe in this model of tau pathology is that these currents are altered in presence of the tau protein. Now, the next step of the project, which will be the object of the next grant application, is to understand why tau proteins lead to the alteration of these waveforms. So, as I was explaining, this is the chamber in which we have placed the brain slice. You can't see it because right now it's sandwiched between the bottom of the plate and the powerful objective of the microscope. So it's kind of a busy scene, yeah? You do have the slice, you do have this probe, which is the temperature probe. You do have this probe, which is the ground, the electrical ground of the electrophysiology probe. You have this probe, which is connected with a glass micropipette filled with internal solution. This is a preamplifier, which then is connected with an amplifier. And here you've got the whole machinery of the two-photon microscope. So in this rig in particular, we can basically measure two main things: the electrical properties of the plasma membrane of the neuron, and also image the structure of the neuron as we have filled it with the internal solution, which contain, and maybe you can see it here, it's a little bit yellowy, a fluorescent probe, which is called Alexa Fluor 488 probe. This is particularly good because it's a hydrazide, which means that it remains trapped inside of the neuron once you have injected it inside. So before we started filming, I've actually prepared an experiment. So this image will not be an image that you see in a camera, will be a reconstructed image, because what my laser will be doing is to scan the sample by exciting it and recording the light that comes out of it. Once we are done with our acquisition, we end up with a stack of images. So you can see here that there is the pipette, and as I move my cursor, each one of these is a different focal plane, yeah? And then as I move down my focal plane, you can see all the cell body of the cell, and you can see the single processes moving throughout the thickness of the different layers. So I can adjust the brightness to make it a little bit brighter. Now that is one level of magnification. Potentially you can go with a much higher magnification. So this is an example of a magnified 4 times neuronal body. So if before you've seen the cell shape and the pipette, now if you go and focus or you actually zoom your scanner onto a process, you can see these are the single branches of the neuron. And if you look carefully, you can see that these branches have these little tips and dots like little spines. And they're called exactly that, spines. And these are the functional compartments where new memories are presumably shaped. And in Alzheimer's disease and in tau pathology, we do have alteration in the structure of these spines, which mirror and to some extent are causative of the electrical changes. So the beauty of this machine is that will allow us to monitor both the structure and the function of the neuron at a fairly high resolution. As we were seeing at the microscope, at the rig, with that machine we can get two kinds of data. Yeah, we can get the electrical activity of the brain, or in this case of the brain slice, related to the network, yeah? So how cells communicate with each other. So these events here called synaptic events or excitatory postsynaptic currents in this case. But also another kind of data that we can get are, the intrinsic electrical properties of the plasma membrane. So for example, with this technique called current clamp, patch clamp, we can monitor the potential of the cell while we inject current steps. So if we inject a positive current step, we can drive the cell to fire these tuck, 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 these electrical spike events, which are typical of nerve cells or of excitable cells in our body. These events are called action potentials, and they work a little bit like an analog digital board. Yeah, they convert. In a way, we are robots which work with these digital events, right? It's the way the cell communicates. Or the information travels across the cell. And also we can measure the currents, the electrical currents that are generated on the plasma membrane to generate the action potentials. So specifically an inward current of sodium ions, these electrically charged particles of sodium known as sodium ions, and the outward current of potassium ions, these electrically charged particles of potassium, which are both needed for the generation of the action potential. And of course, we can compare the properties of all these waveforms between normal mice and mice which have a genetic mutation to express, like in our case, a form of tau that is associated with Alzheimer's disease. And as I was saying, on top of the functional stuff, we can do all sort of a structural evaluation directly onto the brain slice or the cell within the brain slice, so measure the number and the shape of synaptic spines in our neurons. It's just an example of a 1x image as we were seeing at the microscope, and a 4x image. So this is not an optical zoom. This is actually a scanning zoom. Thanks to various techniques of manipulation of the image, we can actually resolve very fine structures. Okay, so our day here at the University of Reading School of Pharmacy is over. We've been through quite a few aspects of what we do here. I hope I convinced you that our techniques are somehow useful to understand what goes wrong in the brain exposed to these proteins, which we know are involved to a certain extent in the onset of dementia. But someone could ask, well, then so what? Once you have seen that these brain slices have an altered synaptic activity or an altered electrical activity, when they are exposed to tau protein or amyloid protein, or they've got weird processes coming out which are not looking exactly as they should, then what? Well, that's actually a very good question, but I think that the essential part of this research is that it allows us to understand what goes wrong and what molecular target is responsible for these changes. It can be a protein. It can be a protein channel in the plasma membrane. It can be even the composition in the phospholipids of the plasma membrane, those fatty sheets that form, the perimeter of the plasma membrane. And once we better understand what is the molecular target that is responsible for these changes, then we have a target for example, a new drug or a new non-pharmacological intervention. Basically, what I'm saying is that for you to be able to develop novel, effective therapeutic strategies for this disorder that right now doesn't have a cure, you need to be able to identify new targets. And the way we do that here is to try to identify targets that are linked to the alteration in the electrical properties and activity of nerve cells.